Every molecular biology technique builds on PCR. If you understand this one method, everything else, qPCR, digital PCR, cloning, diagnostics, becomes so much clearer. So here's the simplest explanation of PCR you'll ever hear. PCR, or polymerase chain reaction, is a method used to amplify a specific DNA fragment, also called the target DNA. To run a PCR reaction, you need just a few key ingredients. DNA template, the sample you want to amplify. Primers, short DNA sequences that define the region of interest. DNA polymerase, the enzyme that builds new DNA strands. Deoxynucleotide triphosphates, the building blocks of DNA. And a buffer solution containing magnesium ions to maintain optimal pH and salt conditions. With these ingredients ready, the PCR can begin. It works by repeating a process called thermal cycling. Step 1. Denaturation. The double-stranded DNA is heated to around 95 degrees Celsius, separating it into two single strands. Step 2. Primer annealing. As the temperature cools, primers bind to their complementary sequences on each DNA strand. Step 3. Extension. DNA polymerase attaches to each primer and synthesizes new strands by adding complementary bases. This cycle is repeated, typically around 40 times. In theory, the amount of DNA doubles with every cycle. However, over time, reagents such as deoxynucleotide triphosphates become depleted, and the reaction eventually reaches a plateau. PCR amplification usually follows three distinct phases. Exponential phase. DNA doubles with each cycle. Linear phase. The reaction begins to slow as reagents become limited. Plateau phase. Amplification stops once essential components are exhausted. So how does PCR work? It heats, cools, and copies, again and again, until tiny amounts of DNA become millions of copies. In practice, that means we can detect and study even trace amounts of genetic material, for example, identifying pathogens or confirming genetic mutations. Now that you understand PCR, you're ready to move into qPCR. Check out our online course, qPCR Setup for Success, if you want structured step-by-step training. Find a link in the description. Don't forget to like and to subscribe for more tutorials.